Welcome, everyone. This is a Research Digest podcast, and I'm your host, Hassan. Cardiovascular diseases are the most common type of non communicable diseases. Among them, congenital heart defects are structural abnormalities of the heart present at birth. And they are most common type of birth defect affecting approximately 1% live births worldwide. But why does this happen to these babies? They do not usually have these typical risk factors for heart diseases. So, why does this happen? Is it genetic or environmental? Today, we'll discuss an article that investigated these questions. But before that, let me introduce you with our guest Talita Choudhury. Welcome to Research Digest podcast.
Hey, thank you so much for having me.
It's my pleasure. First, tell us a little bit about yourself, your career journey, and your current affiliation.
Yes, so as you know, I did my undergraduate in biochemistry and molecular biology at the University of Dhaka. I also did my master's there. And for my master's research, I had actually studied a population of cardiac disease patients, patients suffering from acute coronary syndrome. And that kind of sparked my interest in the cardiovascular field. So I kind of continued my PhD research in that. And I did my PhD at The Ohio State University under Dr. Vidhu Garg. And after my PhD, I am now a staff scientist at the Broad Institute, where I'm also continuing more cardiovascular disease research.
Wonderful. So you're starting from your master thesis, PhD work, and current work, everything aligns so nicely. That's
Yes.
great. Thank you, Talita, for sharing your nice journey and their audience. So the research article that we are going to discuss today is published in JCI Insight, and the title is Impact of Genetic Factors on Antioxidant Rescue of Maternal Diabetes Associated Congenital Heart Diseases. And this journal is published by American Association of Clinical Investigation. And we are happy to have our guest as the first author, Talita. Before we go into your main findings, tell us a little bit about congenital heart diseases, its epidemiology, and why it's important.
Yes. So we all know about adult cardiovascular disease. It is one of the number one killers. But little is actually known about how heart disease affects babies. So congenital heart disease is actually described as structural defects in the heart that are present at birth. So the structural defect can happen anywhere in the heart. So we know that the mammalian heart has four chambers, and the defect can happen in any one of those chambers. It could happen in structures that are present, the valves that are between the chambers. So there is a huge spectrum of defects that can happen under that umbrella of congenital heart defects. Some of them are more severe than others. But in totality, about 1% of births each year are
Right.
affected by CHD, and it is the number one cause of infant mortality.
Mhm.
So it's a very serious disorder. It causes huge economic burden in the pediatric field. And many of these heart defects will affect the patient's entire life. So even after corrective surgery, it significantly impacts their quality of life. They may need further surgeries later down in the road. So it definitely warrants a lot of attention from us researchers.
Right, exactly. So this is pretty serious issue. So before you started this project, what was really known about its relation to maternal diabetes mellitus?
Yes. So, go moving into more development. Congenital heart disease occurs because of an abnormal heart development during embryo stages, and the heart is actually the very first organ to be formed in the embryo. So previously, a lot of research was focused on understanding the genetic defect. So, congenital heart disease, or I'll say CHD from now on. CHD has a very high recurrence rate. So, if one member of the family has CHD, it is likely that subsequent lineage or members of the family will also have CHD. So, the idea was that it is a genetic disease. And previously, research was very focused on understanding what are these genetic risk factors associated with abnormal heart development, and then trying to understand that genetic program that underlies heart development so that we could understand like this risk gene takes part in this developmental program, and just trying to understand that gene regulatory network of heart development so we can try to identify some of these risk factors, these genetic risk factors. However, I think around like the 90s, people started to see that there were some environmental factors that were associated with birth defects. One of the most, I think, common and known ones was a drug called thalidomide that was given to pregnant women to control their pregnancy related nausea.
Okay.
And a lot of these women were having babies with birth defects, specifically congenital heart defects. And that's kind of where the idea that environmental exposures during pregnancy can also cause different birth defects, including CHD, kind of started. Then there were huge population studies trying to identify what are some of these external factors that affect that are associated with high birth defect, especially CHD. And one of those that was very prevalent was maternal diabetes. So if the mom has diabetes, particularly if it's pre-gestational, so before pregnancy, if she has any established type 1 or type 2 diabetes, she has almost a five-fold increased risk of having a baby with CHD. But it was all very epidemiological until very recently. So it was more just looking at association between environment and risk of having CHD. So I think within the last decade or so, we really started digging into what are the kind of molecular mechanisms that are leading to an abnormal heart development due to these environmental factors. And so my research was very much focused on trying to understand these. But I also wanted to bring in how the genetics of the baby can also influence the environmental exposure and how these two factors can sometimes act synergistically to increase the risk of CHD even more. Um, but
Right.
yeah, that's kind of where my research fell.
Got it. So the molecular mechanisms were kind of the main area where it was not touched.
Mm-hmm.
You have kind of mentioned, but still, what were the major knowledge gaps that that prompted you to start this project? Maybe you can include also about the NOTCH1. Why did you target that
Mm-hmm.
particular gene?
Yes, so that's a great question. So again, like there was before kind of the past couple years, there were two very separate fields within the congenital heart defect field. There were people who were studying genetic risk factors, and then there were groups studying environmental risk factors. But we know that in humans, it's never just one. We have a diverse genetic background, and we also have very different environmental exposures during development. So that was kind of a huge gap that people were starting to address in the field. They were saying, like, oh, it's not just one, it has to be both. But no one was able to really show that. So I wanted to look specifically at Notch1 genetic mutations because it is a very, very established risk factor for congenital heart defect. It's been known for over 2, 3 decades now that Notch1 mutations lead to CHD, but it has a very variable penetrance. So not everyone who has a Notch1 mutation will end up with a CHD. So that kind of gives you a clue that there is a secondary factor that's determining it. And I thought, oh, that is most likely another environmental factor. And so I wanted to see what happens if you combine a Notch1 mutation with an environmental factor such as maternal diabetes and see what kind of interactions happen. Does it lead to a much higher incidence of disease, and so on?
Wonderful. Okay, so so let's get into your major findings, right? What is haploinsufficiency, and then your one of the key findings that Notch1 haploinsufficiency and maternal diabetes mellitus is kind of leading to increased CHD penetrance, right? So yeah,
Yeah.
explain a little bit about that.
Yes. So, as I mentioned, Notch1 mutations in humans have been like widely associated with congenital heart defects. So when we talk about mutations in humans, it's slightly different from what we talk about in mice. So I just want to touch on that first. When we talk about mutations in humans, or variation, more like, it can be either a point mutation, it could lead to a deletion of an entire gene. So there's many ways that there could be variation, but overall the effect is there's usually a loss of function of the gene in the humans. So we know that loss of function mutations in Notch
Yeah.
one specifically lead to CHD in humans, or is associated with CHD in humans. We wanted to test this idea in mice, but when you knock out Notch1 completely in mice, so if a mouse just does not have the Notch1 gene, they're completely embryonic lethal, they do not develop, they're not viable because Notch1 is such an important gene. So instead, we used haploinsufficiency of Notch1, which means that the mice only have one good copy of Notch1 and one knockout Notch1 allele. So they're essentially heterozygous for Notch1. And these mice develop normally, are completely fine, until they're exposed to an environmental teratogen, as I will explain. So that's what I mean when I say these mice are haploinsufficient Notch1. They just have one copy of Notch1. So there's almost like a reduced dosage of the Notch1 gene.
Okay.
Yeah. So to move further into the research, what I wanted to see first was, okay, these Notch1 haploinsufficient mice develop normally, right? But what happens when these haploinsufficient embryos are exposed to maternal diabetes? And this was actually done by a previous postdoc, and she published a beautiful paper also in JCI Insight that first described this interaction. So she showed that when you have haploinsufficiency of Notch1 and maternal diabetes, you have a significantly higher incidence of having a congenital heart defect known as ventricular septal defect, so VSD. There's about an 80% incidence of VSD in the Notch1 haploinsufficient mice with maternal diabetes compared to either the non-diabetics, and also compared to mice that have both copies of Notch1, so wild types with maternal diabetes. So
Okay.
these interactions have a much higher incidence than just diabetic embryos and also non-diabetic embryos. Does that make sense? Yeah, so there's like four different groups.
Okay. Okay. Yeah. Yeah. I got it.
Yeah.
So, okay. Another key findings of your from your article is that maternal diabetes kind of impairs proteoglycan distribution in embryonic hearts. Yeah. Tell us about that, and how did you reach this conclusion?
Yeah, so my work kind of spurred from that initial observation where she showed that just having Notch1 haploinsufficiency alone with maternal diabetes can increase the incidence. But I wanted to dig even deeper. I wanted to understand why and where this Notch1 and maternal diabetes interaction was working. So I kind of mentioned that the phenotype that we kept seeing was the VSD, so the ventricular septal defect.
Okay.
Um, and that's basically when the two lower chambers of the heart, the ventricles, there should be a right and left, completely separated. But in our disease model, we saw that there was a big hole between these two chambers, and there was free mixing of the blood due to a septal defect. And I wanted to understand what was Notch1 doing in that structure and why it was so sensitive to diabetes. So I started kind of knocking out Notch1 in each of the cell lineage that was in that septal structure. And we had already known from previous literature that Notch1 is important for endocardial cells. And endocardial cells play a really big part in that specific septal structure. Um, so my initial hypothesis was that if you remove Notch1 specifically in that cell population, you will be able to recapitulate the same phenotype we saw with the global Notch1 haploinsufficiency and maternal diabetes. So that's what I did. I basically deleted one copy of Notch1 in the endocardial cells and then exposed those embryos to maternal diabetes. And sure enough, they again had the exact same phenotype we saw previously. Um, so now that we kind of knew that the Notch1 was playing a big role in the endocardial cells of the heart, of that specific structure, um, that's when I thought of the proteoglycans, because we know that that septal structure, it's really made up of the endocardial cells and then the endocardial cells that become mesenchymal cells and the mesenchymal cells that kind of produce that proteoglycan in that
Okay.
structure. So all of that together kind of makes up that ventricular septum that I was interested in. Um, so that's how then I moved into studying the proteoglycan in that septal structure. And I showed that if you remove not a copy of Notch1 and then expose the embryos to maternal diabetes, eventually it leads to reduced proteoglycan deposition within that septal structure. So that was a key finding that kind of tied together diabetes causes Notch1 deficient endocardial cells to undergo, they undergo mesenchymal cell transformation, but then those mesenchymal cells are no longer able to deposit enough proteoglycan for maturation of that structure. Um, yeah.
Nice. Wonderful findings. Okay. So, the other thing, you in your paper you nicely showed how that Notch1 haploinsufficiency and oxidative stress act kind of synergistically. Okay, tell us a little bit about that.
Yes. So now that I was able to show a direct consequence of what happens when you have maternal diabetes and Notch1 haploinsufficiency, I wanted to move a little more upstream and find out like what what is in this interaction between maternal diabetes and Notch1 haploinsufficiency. And we all know when we talk about diabetes, it is a very big metabolic disorder. There's a lot of different things happening, a lot of biochemical changes. But the number one reason for cells behaving abnormally in a diabetic condition is the associated oxidative stress.
Uh,
Um,
right.
so I wanted to see if that oxidative stress is what was causing the dysfunction in the Notch1 deficient cells. So kind of to test that in an isolated system, um, we actually developed iPSC-derived endothelial cells that also were haploinsufficient for Notch1. So now we have endothelial cells alone that are Notch1 heterozygous.
So
And I'm
it was not done in mice, right?
No, so this part was done in cells because I
Uh,
really wanted to just very specifically see if it was
mm,
oxidative stress alone.
right.
Um, so I was able to get those cell lines. They again were heterozygous for Notch1. And then I exposed these cells to hydrogen peroxide to mimic that oxidative stress environment. And we were able to show, so after I did that, I did bulk RNA sequencing to see what were the genes that were affected by exposure to oxidative stress. And these were compared to just normal wild type cells exposed to oxidative stress. So cells that have both copies of Notch1.
Mm,
And we again showed that the number one affected biological pathway was that proteoglycan and that extracellular matrix deposition pathways. So we were also able to show in a cell-based system that oxidative stress and Notch1 haploinsufficiency interact to disrupt that proteoglycan deposition that we saw in the animal model.
okay. Okay. Wonderful. So these are kind of your major findings. And if for the audience, if you are more interested, please go into the main article. I'll give you a link in the description. Okay, so uh, in this kind of research, we always face different difficulties, different challenges. So tell us about the most challenging part of the project and kind of how did you overcome that thing?
Yeah, so I mentioned the major findings. One of our third finding that was a little controversial, I will say, um, was when we found that oxidative stress impacts proteoglycan deposition, we tried to do a rescue study where we actually introduced an antioxidant into the animal models. So we overexpress the gene SOD1, which is an antioxidant. Um, and we tried to see if having more SOD1 in a Notch1 haploinsufficient background can kind of reduce the incidence of CHD when they were exposed to maternal diabetes. And it was very interesting because when you have both copies of Notch1, so a wild type, with SOD1 overexpression, um, and then they're exposed to maternal diabetes, they do actually lower the incidence of CHD compared to an embryo that had no SOD1 overexpression with
Mm,
maternal diabetes. But when you lose Notch1 and you have SOD1 overexpression, it can no longer reduce the incidence of CHD with
okay.
diabetes compared to um,
Mm,
another wild type. So what we kind of show there is if you have a genetic defect, then some of these antioxidant strategies will no longer work, even if it works in someone who does not have a genetic predisposition. Um, so again, showing there's like a influence of genetic background
mm,
in how you respond to different therapies. Um, So
Yeah.
that was interesting because it's kind of disappointing, right? that
Yeah, so a lot of things are interlinked and it's difficult
mhm.
to get into one conclusion very confidently.
Yes, yeah, because we were able to show all this data showing, oh, look, genetic factors and environmental factors interact. But then at the end, it was like, oh, but the genetic factor still trumps the environmental factor because even when you try to mitigate that environmental factor, you're still going to end up with the CHD because it's so sensitized to the environment. Um,
Yeah.
so that was a challenge. When I actually generated that data very early on in the PhD research, and I was like, oh, this cannot be published because um, at the end it was almost negative data.
Okay.
But even like speaking with the journal editors and the reviewers, everyone actually agreed that this was an important finding, that we have to put it out there. We have to show people that
Mm.
genetic predisposition really matters in the field, and we have to start taking that into consideration when we design therapies. So that was a very, I think, eye-opening experience for
Mm.
me to like look at data in the bigger context of the field. Um,
Mm.
so I would say that was a challenge, but also a triumph.
Interesting.
Yeah.
So sometimes we get negative data or negative results, what we don't anticipate before, but it might be really important at some
Hmm.
point.
Yeah.
So, okay, wonderful. So in your paper, you discussed some limitations of the study, right? So
Hmm.
tell us about some future directions of this project and how can you overcome some of those limitations?
Yeah. So again, we only looked at one gene, right? We looked at
Mm.
notch one, but when we look at a real human baby, they could have variation in
Mm.
several genes, any of which could be a risk for CHD. So I think the next step is to really identify what are those genes that interact with the environment. And the environment could be maternal diabetes, it could be any another big environmental stressor is a maternal um, I guess, hypoxia.
Okay.
Uh, yeah, and even hypothermia, hyperthermia, and then there's several drug use that are affected. So I think it would be important to see what are those genes that are more sensitive to the environmental factor, because then we could start screening for these genes and see which I guess populations are more sensitive, and then we could actually design either therapies or
Mm.
lifestyle changes for the pregnant mom to make sure that these sensitive populations are more protected. Um, so I think the next step should be trying to identify other
Mm.
genes that interact with environment, and some of the limitations in my work, I would say, is we only tried out one antioxidant therapy. So we were looking at SOD1. But I think even in that notch one maternal diabetes interaction, we can like explore further to see if there are other ways we could reduce the incidence within this gene environment interaction. So yeah, I would suggest to the next person to maybe look into that.
Wonderful. So finally, do you have any message to our audience? Anything on career or science?
I think my biggest message would be do what you love. Chase important questions, even if they're hard. There's going to be an assay that will help answer your question, and if there isn't, then develop your own assay. Um, but just go with your heart. Whatever excites you, do it.
Wonderful. So, Talita, thank you so much for coming to this platform and sharing your wonderful research.
Thank you. Thank you
And
so
thank
much for
you.
having me.
Okay, you are welcome. Thank you, audience, for listening to us. We'll see you in the next episode. Bye.
Bye.